And oops. So from last week, we close but didn't quite finish off where we were supposed to be with our previous lecture. So we will quickly finish off the last section, which was about principles of diagnosis, and move from there on to the last section or the last lecture I've got on diagnostic tools and principles, where we're going to talk a little bit more about some of the laboratory-based tools, molecular tools, serological tools, etc., that we can use to help identify pests and pathogens. You won't get to experience these in the prac classes, but to understand what potential tools there are. So, taking a step back, if you recall from last week, we were talking about, or in summary, we're talking about symptomology, and we're talking about recognition of symptoms in the field. We're talking about the knowledge that symptomology is important for diagnosis of disease and disease management. But we also emphasised the fact that we really have this dynamic association between your pathogen, your host, and the environment, and that can, as we discussed in terms of disease onset, that can also vary your symptom expression. So variation in your pathogen may vary the type of symptom you get. Variation in your host might vary the interaction between the pathogen and host, and therefore the type of symptom you get. And of course, environmental factors and time, for that matter, will also vary the potential severity of the disease and otherwise. So all of those factors become important when you're considering your observations of symptomology. What I want to do to finish that section off was really starting to do a little bit what we sort of covered off on that virtual plant pathology exercise, where if you remember, the very first part of that is you go out into the field and you start talking to growers and you start getting some clues from the source of the disease itself. And these become quite important. These can be absolutely critical for identifying what is the actual cause behind a particular disease. So firstly, it's really important that you look holistically at the whole field. Don't just focus straight on your diseased plant and focus on that and that's it. You really need to look for patterns, look for clues. You are the plant detective. You're dealing with a murder scene. In this case, it's a plant that's died or dying, and you're trying to find out all of the clues as to, you're doing the whodunit, but it's not just the pathogen. You want to find out what is the situation that led to this problem. Are there patterns, distribution patterns? Maybe it's clumped, maybe it's random, maybe it's around the edge of the field. All of those can indicate the type of disease you're dealing with, but also the source of it. You know, say for instance here's your field, you've got a predominant wind in that sort of direction, and all your diseased plants are sort of, you've got this gradation of high level of disease and lower, and that sort of decreases as you move in. What is that sort of telling you about where the pathogen's coming from and how it spreads? Or at least suggesting. Yeah, it's coming from somewhere up here. And it could be another crop, it could be a bunch of weeds. It's probably not coming from the seed. If it's coming from the seed, what sort of pattern might you expect it to have? It'll be probably random, because it's scattered randomly throughout the whole crop because you're randomly sowing the seed. And it's probably windborne because it's moving in that general direction with the wind. Alternatively, you know, you've got a field and you've got a big patch of disease in the middle of it. What might that suggest in terms of how it spreads? In to me it suggests, and not guarantee, but to me it suggests it might be moving fairly slowly. It might be moving within the soil system. It's probably not airborne because it might go from there and go all over the place. It's probably moving plant to plant to plant in an expanding patch. Or alternatively, maybe look at the geography, and is that an area where you've got poor drainage? Maybe it's a hollow. Maybe you've actually got a waterlogged site in that point, and that is creating an environment that encourages moist soil, anaerobic soil conditions, and leading to disease. So looking at the patterns can be really critical. I don't know if you guys can see that. I can't see it from here, I must admit. Looking at the patterns can be really critical in terms of describing, of giving you clues as to what it is you might be looking for, but also how it occurred. So you want to look at things like the soil type. It may be associated with the type of soil you're dealing with. The topography, is it a hollowed area? Is it a north-facing slope? Is it warm? Is it cool? Likely to be. Is there a hard pan? Is the soil structure poor? And that might be leading to poor, stressed plants. Is the pH out of whack? Or some pathogens, for instance, like alkaline soil conditions, some like acid soil conditions. If you've got some knowledge of what the conditions are, that might also suggest why the environment is actually conducive for disease. Farm traffic. Why is farm traffic important?
Yep. Mechanically.
Oh, yep. And
yeah,
yep.
that's what I was
Yep.
going to say.
Potentially spreading it mechanically, so if it could be carrying the pathogen with it. It can also be actually damaging the crop. Could be compressing the soil. It might be down the spray runs where you've actually got compacted soil, you've got stressed plants. They're actually more susceptible because they're just not healthy in general, and more susceptible to the actual pathogen that's a relatively weak pathogen, but it takes out weakened plants, for instance. So all of those things can be important. Is there a weedy crop margin? Maybe that's an inoculum source, or even a source of vectors, wind direction, etc. Here's an example. So we've got a nice patch here. In this instance, this particular disease was spreading, I think, by a rain splash. So rain comes down, hits the leaf, the spores kind of get blasted off, but they can only go, to neighbouring plants. They're not blowing miles away. So you've got these initial spots that may have been started by infected seed, or may have just been introduced there in some manner, but then it's slowly spreading plant by plant, and you're getting these patches. Discuss the problem with the grower. Again, in our exercise, on that exercise, they were emphasising that too. This can be, again, really critical for finding some of those clues. Now, what sort of pesticides were used, if any? It might have been an inappropriate pesticide. It might have used the wrong rates, might have used the wrong materials. So that actually might be the problem. What sort of fertilisers? What was the seed source? What was the seed held? Was it certified seed? Was it tested? Were there any seed treatments? Seed source, maybe farmer X has got a disease, and then you find out farmer Y has got a similar one, and they use the same seed source. It's starting to indicate where the problem might be coming from. What is their rotation practices? What are the factors that could potentially have built up pathogen inoculum in their system? The disease history, have they seen this before or not? Has it? Ask them, has it seen it? Have they seen it spread? You're only coming into the crop, and if you're an advisor, you're coming to the crop once and you're seeing it in one point in time. The grower would presumably have seen it as it's grown or as it's developed over time. So, is it spreading? Have they seen it before? Is it getting more or less? Is it just a bang, it's happened and nothing more is happening? Or did it start in that corner and out slowly spreading, and they can provide some of that clues? What's the weather been like? Is it cold and wet, hot and dry? Has there been a drought? Has there been a lightning strike? There's a classic example here. This was I was saying how you need to be in the business for about 30 odd years before you're an expert diagnostician in a crop. Probably the best potato diagnostician the state's had for a long while. After 30 odd years working with potatoes, walked into a crop and he saw something for the first time going, I have no idea what the heck this is. and there was the plants were all flattened, and you pull them apart and they're necrotic right through the centre of the stem, but no obvious damage apart from that. And you sit and go, I have no idea, I've never seen that before. Turned out it was a lightning strike. So lightning had smacked in the middle of the potato crop, cooked all the stems, killed all the plants obviously. Not a frequent event, but one by doing a little bit of due diligence discovered the cause. And again, some of the agronomic practices. Is it where they're turning the tractor and they're actually accidentally spraying those plants twice because they've sprayed it on that run, they've turned around and they sprayed it on the way back by mistake or something? Finding stuff from the field is important. Then, hitting the literature, because most diseases, as we mentioned, will have been dealt with in some manner before and will be described. So there's a lot of good sources of reference sources for diseases in crop plants. There can be guidebooks. I forgot to bring one down. You get these little ute guide sort of things that have nice pictures of common symptoms, and that can be really valuable to flick through and actually look at. Well, that looks a bit like that. That gives me some clues as to what it might be. Now I'll try and confirm it. Texts, scientific articles if it's something fairly newish, or grower guidebooks. Internet can be useful. You just need to be pretty careful with non-refereed sources that are actually accurate. Some of those websites kind of suggest things, use images that might not be correct, but they can be useful guides. And if you are going to use those sorts of sources, choosing government ones or university extension programs is usually a bit safer than picking a best grower guide from a nursery or something. Okay, so just to finish that component off, to be a good disease diagnostician, you really are being a detective. You're trying to use a whole range of information to find out what is the cause. Symptomology is useful, but you also want to find out patterns in the field. You need to find out that background information about how the crop was grown, careful field observations, and literature searches and literature information about, okay, this disease looks a bit like this. What are its conducive conditions? Oh, like cold and wet. Yep, that ticks off. We had cold and wet. We had a disease that looks a bit like that. We have a susceptible crop. Where the rotation sort of suggests that might have made it worse. Okay, now I've got a lot of clues to suggest that this might have been caused by this particular disease. Now I take it to the lab and try and confirm it. So doing that background stuff before you go to the lab, which is what we're going to do in our next prac class, and start ripping pathogens out of diseased plants. That background due diligence is really important. Okay, so that was finishing off last week. Any questions before we go on to what I'm actually supposed to cover today? Right, so we've gone from discovering a disease, noting the symptoms, trying to work out, characterise that symptoms, that looks a little bit like this particular disease, getting that background field information. We've gone and taken our samples, and we're now trying to test those samples for the presence of a pathogen. And what sort of tools can we use? Well, first, it's really critical that you get good samples. Having dried, dead material may make it very difficult to try and find anything in your material. So you want good representatives of the diseased plants. You want some healthy plants for comparison, ideally, if you can. You may not want them too far gone. So if the disease has got to an extreme end, and things are really starting to rot badly, why might that be a problem? Why do you think if you've got a plant that's really diseased, just about dead, why might that be a problem for trying to find the causal pathogen? Any thoughts? and it's something you're going to discover in your next prac class. The pathogen might initiate the disease, but there's a hell of a lot of other microbes out there that like dead stuff and eat dead stuff. So the pathogen, if you've got a living plant, there's only very few microbes that have the capacity to eat living plants, and they're called pathogens. So they infect and they start to cause disease, and they might start to cause necrosis, they might start to cause cell death. There's an awful lot of microbes that can eat dead stuff. So once that material has started to die, all these other microbes can come along and start to feed on and decay the dying plant tissues. So if you grab a plant sample that's very decayed, like very diseased and almost dead, the pathogen should still be present, one would hope, but you'll also have a lot of other contaminating microbes that are eating the dead material. So if you're trying to find the cause and not all these associated microbes, you're now looking a needle in a haystack to some extent. You'll have a lot of other microbes to try and sift out of the way to find the real one. If you get something that's a little bit earlier in the process, where the infection's started a bit earlier and the disease is just starting to express, it's more likely that the pathogen is present with a lot less of these contaminating microbes. So collect appropriate samples. If you suspect it's a soil-borne disease, so you've looked at the plant, you've noted actually it's got rotten roots, or it's got lesions on its roots, looks like it might be a root pathogen. You may need to collect soil samples too. If you're looking for nematodes, for instance, they might be residing within the soil and not necessarily easily found on the root material itself. When you've collected good samples, make sure you look after them. So don't throw them in the back of your car on the back windscreen and let them bake. Stick them usually in a plastic bag to maintain humidity, maintain moisture so they don't dehydrate. Avoid excessive heat. So if you've got a esky or something, you can toss it in those. Get them back to the place where you're going to process them as soon as you can. Get them into a cool environment before processing. Obviously don't freeze them. Okay, so next step. So we've got our good samples. We now want to actually start to check to see what is it that we've got. And you'll see this is mirroring a lot of that exercises you did in that virtual plant pathology exercise, which is I guess the major process we're trying to follow. Look at the plant samples first. A common mistake that some diagnosticians might do is you've got your plant disease and you immediately try and plate it onto an agar plate and see what grows on it. You're probably going to find more information by directly looking at the plant sample itself, where the pathogen might be present. So low and high powered microscopic analysis can be useful. You might be able to see signs of the pathogen, maybe sporulation, a bit like those mildew samples we had in the prac class last week. Or take it up to a high-powered magnification, your compound microscope, and actually start to look for presence of bacterial cells, presence of fungal mycelia, presence of nematodes perhaps, even insects or the likes. So if you can start to see some of these structures, and we'll talk a little bit more about those as we go on to the different pathogenic agents a little later. But that then can give you some real clues of what you're dealing with. If you see a huge network of hyphae and some sporulating structures, you're going to start to suspect you're dealing with a fungus and not a nematode or not a bacteria. If you've got billions of bacterial cells oozing out, you're going to start suspecting you're dealing with the bacteria. And that decision is important because if you do come to isolate, you need to choose the appropriate media to do so. You don't isolate a fungus on a bacterial media, for example. And even the absence of something can be really important. If you're dealing with a viral disease, for example, you're not going to see it with a compound microscope. A low and high powered microscope is not sufficient to actually visualize a virus. So you might see yellow chlorotic leaves, you look down the microscope, you see nothing. You don't see any presence of a pathogen. That might suggest to you, A, it's potentially a virus, or B, what would the other thing it could be? Yellowed leaves and no pathogen present. Obviously pathogen present.
Nutrient deficiency.
A nutrient deficiency. So you got yellowed leaves, gee, could it be a nutrient or could it be a virus? Then you can maybe go and do additional virus tests, for example, and see if it's that. So, when we talk about these different tests, we're going to talk about the good and the bad bits about them. The pros of your direct microscopic examination is you get direct evidence of presence of the pathogen, so you actually see it. And that can be quite valuable in terms of testing, as opposed to some of these other tests we're going to talk about, which are indirect tests. You run a DNA test, and you're hoping that it was actually the pathogen and not some contaminant flying around in the lab that you actually detected. This second component is also really important. A number of plant pathogens are unculturable, so which means you can't extract them out of the plant and grow them on an agar plate. They won't grow. So your mildews, for example, are what we call obligate parasites, obligate pathogens. They can't grow on agar plates. So if you went straight from diseased plant sample to agar plate, you're going to miss it. It won't be there. Direct observations of their leaf, you're definitely going to find it because it's pretty obvious, and you're going to see the presence of the fungus growing quite happily on the leaf. So detection of unculturable organisms, really quite important. The other thing is you're not only directly seeing the pathogen, you're also associating it with the symptom. So rather than isolating and then finding something and growing out and characterising that, you're actually saying, I see this mycelia and it's associated with the necrotic part of the leaf, not the healthy part. So it really does seem to be associated with the disease I'm seeing. The negative, look, there might be other organisms, contaminated organisms, there might be spores of saprophytes that aren't pathogenic sitting on the surface that you might see, and it could confuse you. Hopefully they're not in high abundance, but there is that chance. If it's a really messy, horrible sample, as we were discussing before, there might be lots of these microbes present that aren't pathogens, but they just happen to be enjoying the ride and eating all the dead material. It's also relatively low sensitivity, so it depends on your capacity to find the stuff versus some of these more sensitive tests we're going to talk about in a minute. So, it is important. It has some limitations. What other microscopy can we do? Well, for viruses, phytoplasmas, these guys are too small to see with normal light microscopes, your compound microscopes. We can go up a stage and go to what is an electron microscope. I don't have a picture of an electron microscope, but they're big fancy machines, expensive machines. Essentially they operate pretty much the same way as a normal microscope, except they fire electrons rather than light at the sample, and the resolution's much higher. So instead of being, we were talking about your compound light microscope where you've got a 1,000 times objective with a 10 times at the top, so you maybe get up to 10,000 times, I think, is that right? Or is that, no, sorry, it's 100 times by 10, so you get 1,000 times magnification. That's the best you can sort of get. These guys will go 100,000 times. And you can see things that are sub-microscopic. So that's a virus particle, that's a purified preparation of virus particle. You see all these nice little spherical things. They're all individual particles. So if you've got a suspected virus, you could do just a crude sap extraction, stain it, have a look under an electron microscope and see if you can see particles. And that is a diagnostic tool that is used frequently. The main virus diagnostic lab in town, they run a service particularly for a range of viruses, but for any orchid samples they get in, orchid fanciers around the country send their orchids in to get virus tested. They're all done by electron microscopy because those viruses are quite easy to find under electron microscopy. So they just take some leaf, grind it up a little bit, add a heavy metal stain. The heavy metal stain sort of makes this contrast colour. and look down the microscope and find round ones or squiggly ones or different shaped viruses, and then they know it's present or not. Phytoplasmas, these are these prokaryotes. They're a bit bigger than a virus, but they're still pretty small. You can, if you suspect one, you can do, and this is a section through an infected plant, and this is the phloem, and you see these round cells, which is indicative of the phytoplasma. I didn't do a pros and cons. What's a pros? Obviously, high magnification allows you to detect things like viruses, or allows you to directly observe viruses. So if you're doing a virus test, if you can see it, gee, that's good evidence that you've got it there, as opposed to some of these indirect tests. The negative, it's relatively insensitive. So just because you don't find it, it might be a low concentration viral pathogen. Or alternatively, this is a purified virus sample. A normal plant sap sample, you'll have lots of squishy cell membranes and round globules and all sorts of other things that are present just from normal plant, ground up plant cells, which can confuse you if you're not terribly experienced at trying to find the difference between those and a virus particle itself. So sensitivity is the main negative. Right. So the next, we've observed the plant samples. We've done what we can to actually see what we can see in the plant sample itself. We can now then go to the stage of trying to isolate it from the plant sample onto an artificial medium, and then once you've got it in an artificial medium, you can actually grow it up and actually study that pathogen in more detail. The choice of the medium is important. You can have a generalized medium, a selective medium, or a diagnostic medium. Okay guys, what's the difference? What's a general medium, selective medium, and a diagnostic medium? General medium, hands up.
It'd be used for all different types of diseases.
Yeah. So something that's not that's going to allow most things to grow. There isn't a perfect media that's going to allow everything. Well, I mean, there's some organisms that won't grow at all anyway, but there are some general things like nutrient agar is pretty good at growing most bacteria. Not so great with fungi. Instead, something like potato dextrose agar is pretty good at growing most fungi. So that would be a general fungal or a general bacterial media. What about a selective media? What's a selective media? I once put this in a exam. What's the difference? And the best answer I ever got was someone who said, "It's changing the channel." Selective media. But a selective media. So we've got an agar plate, and we want to isolate a particular type of pathogen. So we can put in, for instance, antibiotics into that media, and that will actually kill off most of the bacteria. So if we know there's a lot of bacteria contamination, we're trying to isolate say a water mold, we're trying to isolate a Anuomycete fungus. That's relatively hard to isolate, and we know there's lots of other bacteria present. We can use a media that with antibiotics in it to try and kill all those bacteria off and encourage the isolation of the particular fungal pathogen. We might even look at different nutrient sources to actually specifically select for a type of pathogen that we're after. So if from our background work we've looked at the symptomology, we think it might be a Phytophthora. We think it's this type of fungus. It's invading the roots. We're pretty sure it's in that area. We just want to make sure. So we want to make sure we isolate a Phytophthora. We don't isolate, some other rubbish. So we'd go and use a Phytophthora-specific media to isolate that, and then try and confirm that the pathogen we suspect is the one that's actually there. Diagnostic media. What's a diagnostic media as opposed to a selective media?
might show something up in a different colour or
Yep.
shape.
Yeah. So it may not stop other things growing, but when the thing you want grows, it might provide a colour change or do something. And then, a classic example, one you'll probably have a go at with in the prac class, is a medium called King's Bee medium, which is general in some ways. It allows lots of bacteria to grow, but if you've got a fluorescent pseudomonad species, on King's Bee agar it produces a fluorescent compound that under ultraviolet light glows. So if you want a quick test, if you're suspecting it, and there are a number of fluorescent pseudomonads that cause plant disease, so if you're suspecting, I think this could be a Pseudomonas species affecting my beans, I've done an isolation. I've got a whole bunch of white colonies. Well, they'll look the same. All white bacterial colonies kind of look pretty similar. But I've grown it on King's Bee media. I've tossed it under the UV light and it's glowing. I strongly suspect I'm dealing with a fluorescent pseudomonad. And then you can do other tests to confirm that. Whoop. So what are the advantages of isolating? It allows you to study that organism in pure culture. So get rid of all the other confounding microbes, get rid of the plant material itself that might be obscuring what you're trying to see, bulk it up into large amounts so you can actually look at it more closely. You can study its morphology. It might be that not everything is present on the plant itself, but maybe once you've grown it, you can actually look at the mycelial structures, and you might encourage it to sporulate and look at the spores. Maybe the spores weren't present on the plant sample. And for fungi, spore structure is often the most diagnostic morphological characteristic. So you kind of want it to sporulate to try and identify what you're dealing with. Once you've got it isolated, you can do things like nutrient tests, biochemical tests. So you can grow up this biolog. That's a bacterial assay where you grow it up on a whole range of different carbon sources. And if it grows or it doesn't grow, that'll give you an indication of what species you're dealing with, because that's been characterized. The other really important thing you can do is if you've isolated your pathogen and you really want to say, look, it looks like a pathogen, it sounds like a pathogen, it smells like a pathogen. Is it a pathogen? I'm going to take it and I'm going to inoculate a healthy plant, and if it causes my disease, I'm now pretty damn convinced that this is the pathogen. And as I was saying, if all you see is mycelia on the leaf, you go, it's a fungus, but I don't know which one. You can isolate it, encourage sporulation, and then say, well, now I know it's not just a fungus, it's a fusarium because it's producing fusarium-type spores. And we'll discuss that more when we get onto the individual pathogens. What are the negatives? We already mentioned this. So if you're dealing with an unculturable pathogen, well, it's not going to work. So if you're dealing with a virus, you can't grow a virus on an agar plate. Ain't going to happen. If you're dealing with a mildew, powdery or downy, it's not going to happen. If you're dealing with a rust fungus, it's not going to happen. Dealing with a phytoplasma, it's not going to work. So there's a whole range of pathogens, that nematodes as well, that can't grow on agar plates. A lot of them do, but there are some that do not, and therefore this technique is not going to be useful. You may be dealing with a pathogen that's relatively difficult to isolate, and or when it does isolate, it grows fairly slowly compared to the environmental microbes present. So it might be, even if you do successfully isolate it, the plate might be overgrown by all these contaminant microbes that grow really fast. So maybe a trichoderma comes onto your plate and just grows, and the whole plate goes green, and you go, well, that doesn't help because I know that's a contaminant, it's not a pathogen. I don't know what my pathogen is because it's now been overrun by this fast-growing contaminant. Similarly with bacteria, maybe you're trying to isolate a particular slow-growing bacteria, but it gets completely flooded with bacillus that's not terribly relevant for the disease. there's no perfect medium, so you need to make some choice of your right type of medium, whether that's a generalised, but even the generalised one, you're selecting a medium that's going to select for predominantly bacteria because you suspect it's bacterial, or fungi because you suspect it's fungal. But if you made that call wrong to start with, you might not get the pathogen out in the first place. And similarly, selective or otherwise. You might actually find, we're talking about getting, if you see a few mycelia on a leaf and getting out to a plate, and then you can encourage sporulation, might help identifying that particular culture. Sometimes it can be the reverse too. It might be on the leaf itself you've got sporulating structures. Once you get on an agar plate, the thing refuses to do anything or won't sporulate. Some fungi actually don't have obvious spore structures, and certainly not on agar plates. So you end up with a bunch of mycelia, this white fluffy stuff, and that's not going to be terribly diagnostic just morphologically, maybe. So that can be a problem. The other aspect is you are isolating a pathogen and you're separating it from everything else so you can study it. There are some diseases, at least, which are actually interaction between more than one pathogen. So the symptom you're seeing is either additive from two pathogens working together, or you actually do physically need both pathogens to actually cause the disease. And so once you've separated them and you study one half of the equation, you're missing the other half, and so you may struggle to really understand what's going on. Best example I can think of there are actually not ones you got on agar plates, they're viral ones, but we'll come to that when we talk about viruses in about 20 minutes. That's isolating onto agar plates. You can also try and isolate different pathogens by different means. If you've got infested soils, some phytophthora, you can use baiting systems. So by baiting is you stick a susceptible plant, or sometimes an apple or something else. You can actually take what you suspect is the soil that's got the pathogen in it, and you either plant a little plant in it, let it grow a little bit, and see if it gets infected, and then you can confirm that soil sample has got the pathogen I suspected in it. By an indirect method, by planting a healthy little plant in that soil, or another piece of fruit or something, and then isolating from that bait plant or the bait organism. If you've got a nematode in the soil, again, obviously you can't grow them on an agar plate, but you can actually sieve the soil for presence of nematodes. This is what we call a Bayerman funnel. So here you have your soil, you mix it up with water, you have a little filter, and you leave it there with a light on the top, and the nematodes sort of wiggle their way down and eventually fall down and accumulate in the bottom of there. You can squirt that out into a slide and have a look and see how many nematodes you've got and what they look like. So that can be a useful tool for determining if you've got pathogenic nematodes, plant pathogenic nematodes in your soil sample. And we're going to talk about what that actually means. For certain fungi that have large resting structures, these sclerotia, they are resting structures which are really just big melanised lumps of mycelium, and they can be sort of 1 or 2 millimetres. Some of them can be 3 or 4 or 5 millimetres. They actually can be moderate size. You might be able to sieve through some soil using differential sieves, get rid of the big stuff, get rid of the small stuff, look for the things that are about the right size. Are there any sclerotia present? And then you've got a reasonable idea, yes, okay, I'm dealing with that. I thought it was that pathogen, and I've found plenty of it in the soil. Those are mostly soiled ones. You can also look for presence of the pathogen moving through the environment in the air by having spore trapping. So if you put up, generally they are sort of like sticky traps. As the air passes through the actual trap, the spores get deposited on the surface, and then you can actually look at that and observe presence of particular spores that might be associated with the epidemic that you're actually seeing in the crop. There we go. Ooh, we're going to run out of time again. Of course we are. What else can we do? We can. There are a number of other tests we can actually try. If we suspect it's more nutritional, obviously there are nutrient analyses of soil and plant tissues. That might be if it's a directly nutritional disorder, or it might actually be useful if that's creating a conducive condition for a relatively weak pathogen to come and do its job. So it might be, you can see there's a nutrient imbalance that's actually led to a weakened plant that's allowed a fungus to take over. We can also use serology, which we're going to talk about in a minute, or nucleic acid based tests, which are very valuable assays for specifically identifying individual pathogen species. More indirect tests. Serology. Excuse me. Serology is using antibodies. So, our understanding of how antibodies work and how antibodies fight infections, or at least recognise foreign things in our bodies, has realized, people have realised we can use this as a diagnostic tool to identify things. Because each antibody specifically recognises a particular antigen, so a structure on a pathogen or on a protein that actually induces a response in an animal that then produces this antibody. So the antibody will specifically recognise a antigen. So how do you make them? Very crudely. Well, actually, we'll go on to that first. Animals obviously have this quite sophisticated system for fighting microbial enemies, these antibodies, these proteins. They recognise the foreign molecules, they bind to it, they neutralise it, they get rid of it. So antibodies are a very important part of our and other animal defences. They're not in plants, of course. But we can use them because they specifically recognise and bind to, like a bacterial pathogen in our system. We can use that capacity for its specific binding to actually identify the pathogens of interest in plants. This is probably most widely used for virus detection. One, because it's very hard to, obviously microscopy is pretty useless. It's pretty hard to detect them otherwise, but they're also really well designed for this, because a virus is a relatively simple structure, has got essentially one major protein comprising its structure, and therefore is very good at producing specific antibodies against that virus. But it can be used for bacteria, fungi, nematodes, all sorts of other things. It is no good. It doesn't detect things that the animal thinks are normal. So it will not detect nucleic acids. Nucleic acid is, that's something we expect to find. If it's a foreign protein, it'll produce an antibody to it. It won't produce an antibody to a nucleic acid. So nucleic acid-based pathogens like viroids, you can't use serology against. So, what is an antibody? They are otherwise known as immunoglobulins. They are proteins, or a collection of proteins. So it's usually a protein complex, and often they'll bind together. You have this conserved region here, they'll bind, and you have more variable regions here. And this part of the protein is where it binds to and recognises the foreign thing, the foreign protein, the bacteria, the virus, whatever. The structure, well, you can read that there at your leisure. It's not that critical at this point. For diagnosis, it's important to know you can actually get two types of antibody, or two groups of antibody that are useful. One is polyclonal antibodies. These are, if you inject a rabbit with your virus pathogen and it produces an immune response, it produces a family of antibodies. So while you've got a virus that's got a fairly simple structure. There's probably an antibody that recognises that part of the protein, another one recognises that part, another one there, another one there, another one there, another one there. Maybe some of the contaminant plant parts that you've accidentally, when you've purified your plant samples, a few other plant proteins. So you end up with a family of antibodies, not a single antibody. Now, most of them might recognise the thing you're after, which is fine. So it can still be very useful as a detection tool at just detecting multiple epitopes, multiple parts of multiple antigenic sites. And that's called a polyclonal. A monoclonal is where you go, I want to be a bit more specific. I just want that one. I don't want the others. I just want the one that recognises that particular protein. And I want it that specific so I can recognise that particular strain. Because maybe my polyclonals for, when I inject my bacteria into the system, I get some specific for that bacteria, but lots of them are generic for bacteria in general. And so it hasn't been terribly, my polyclonal antibody hasn't been terribly useful as a diagnostic tool because it detects all of the Pseudomonads. I want the one that detects Pseudomonas species X. So I need one antibody, not the family of antibodies. So how do you do that? If you look at the way antibodies are produced in animal systems, they're actually made in the lymphocyte cells. So each individual beta lymphocyte cell produces one antibody. Of course, there's lots of cells, and so that's how you get the family of antibodies. But if you immunise, it's usually in rats, immunise a rat, isolate the spleen where the lymphocytes are, dissolve all those cells up and culture each cell individually in cell culture, you'll be producing an individual antibody into those cells. Then you have to screen the thousands of cell lines and find out the one that actually produces the antibody you really want. But once you've got it, you've now got a monoclonal antibody, a single antibody that detects the thing you actually want and not the other family. So much harder to make, much more expensive to make, but once you've got it, it's really good. That's what I just said. Hard to make, some advantages. One advantage, you get homogeneity of the antibodies, so you get uniform results. Polyclonal antibodies, you have to keep making them. You have to keep purifying your whatever, injecting into an animal, bleeding the animal, purifying from the blood. A monoclonal antibody, once you've got that cell culture, that's immortal, and you can culture that cell forever and produce that antibody forever. So you'll have the same antibody produced once you've actually produced it. In virtual unlimited supply through multiple cell lines. Actually, it's kind of a bit sad. They actually introduce a cancer into the rat, which is why you get this immortalized cell lines. But we don't know about that. It can also be highly specific because it's a single epitope instead of multiple epitopes. It's also because you're getting a single epitope. If you're trying to introduce, trying to get an antibody to a virus, for instance, purifying viruses is not the easiest thing in the world. You're grinding up plants, you're trying to purify the virus, but you're probably going to get some plant proteins co-purifying with it. They will also be antigenic in the animal system you're injecting it into. Going for a monoclonal, you're selecting the ones that actually only detect the virus, so you actually remove all of those antibodies that might be detecting plant stuff. Why would an antibody that detects plant proteins be a real problem if you're trying to detect a virus? So you've got an infected plant, it's got a virus in it, you've got your antibody, a polyclonal antibody that's got mostly virus antibodies, but it's got some that also detect plant proteins. What if it's a healthy plant? Are you going to get a positive result? Maybe. It depends on the contaminant level. If there's plant proteins that react to antibody, you might actually get this high background of, I actually recognise bits of the healthy plant as well as the diseased plant. So that can be problematic. We're going to talk about that soon, maybe in the next lecture by the sound of it. The monoclonal antibodies are also really useful for high sensitivity, high affinity assays, such as lateral flow devices. What's the most common lateral flow device? Pregnancy test kit. So, you know, you put your sample in and it runs through and you get 1 stripe or 2 stripes, you're pregnant or you're not pregnant. That's a monoclonal antibody against, I don't know if it's against estrogen or something, I'm not sure actually what it's against, but indicating whether you're pregnant or not. You can do exactly the same thing, and there are exactly the same thing for plant pathogens, where you add your plant sap sample on the end of one of these lateral flow devices, it runs through, you get a band, or you get 2 bands. 2 bands means you're infected, 1 band means you're healthy. The same technology and can be very useful for in-field diagnosis. They can be negatives, monoclonals. They can be too sensitive, and sometimes you want something that'll detect all of these. You know, there might be multiple strains of the pathogen, but you really want to detect all of them, and your monoclonal might only detect some but not all. It might be too specific. So you need to be careful about your choice of monoclonal. We're running out of time, aren't we? Why don't we have a break at this point? And yeah, we'll have a break, but we'll get rolling smack on 12 as according to that clock, not according to my watch. So see you in 5 minutes or so.